Hello dear learners, in our today's video we are going to learn about the mechanism of DNA replication in prokaryotes. So in our last video we had already talked about that how do the DNA polymerase work, and we had talked about the structure of DNA polymerase 3. Now this DNA polymerase 3 is the main enzyme which will carry on the process of replication in the prokaryotes. And we have already talked about that the DNA polymerase 3 works better than DNA polymerase 1. But then in prokaryotes replication, somewhere in the process, we will also require the DNA polymerase 1. Now, one thing you need to know is that the type of DNA which the prokaryotes have is circular. And this is a very well known fact that prokaryotes do have a circular DNA. Suppose this is the DNA of prokaryotes, and it is double stranded, and for the sake of simplicity I have separated the two strands so that you can know it in a better way. Now always remember that there is one ORIC is that region where or from where the replication will begin in the prokaryotes. And always remember that the replication in prokaryotes is bidirectional, means it can occur from here also and here also. So overall, if we want to make the directions of the replication and how these things happen, so let's take this microscopic view of this part. So it is like this. In this strand, please remember that this is your 5' to 3' end, and this is your 3' to 5' end. And suppose here is your ORIC, origin of replication initiation complex. Now the DNA polymerase has to work, that is DNA polymerase 3 has to work over here. Always remember that DNA polymerase 3 will work in the direction of 5' to 3'. That means it will do its polymerization activity in 5' to 3'. Means it has to recognize the 3' to 5' strand, or it has to move in this way, then only it can do the polymerization. Means what I mean to say is that if this is your 5' and if I have divided this whole strand, this strand from this ORIC, then this becomes your new 3' and this becomes your new 5'. So now here in this strand what happens is that the DNA polymerase will work very simply. It has to just move from 3' to 5'. So if here is the primer, I will tell you about this primer and who will recruit this primer. I will also talk about that in detail. But please remember that after this primer, here from here only the DNA polymerase 3 will work in the very continuous way. It will continue doing the replication very simply. So here it becomes the leading strand. Here the case is different because at a time what happens is that only one of the replication fork is open, not whole of the DNA is not open at the same time. No, so only a part of DNA is open, and that time only that part of DNA can be replicated. So suppose if this is only the part of DNA which is open, and then afterwards let's say that it is closed from here like this. Suppose it is closed. So can you think of any idea that the DNA polymerase can start working from the, let's say, this region, and then it starts moving here? Do you have any idea? Like, can it move like this way? No, it will not move unless this replication fork further does not open. The DNA polymerase cannot continuously synthesize this. And why it has to synthesize this way? Because here it is the 3' and here it is the 5'. So now what happens here? The lagging strands start to form, and the lagging strands are in the way that first the primer will form here, and then up till here the polymerization activity will take place. The new primer here, then here the polymerization activity will take place. So this is the condition of the lagging strand which is there because of the DNA polymerase 3. So on the half part of that same strand, the leading strand will be formed, and the other half part, the lagging strand will be formed, and just the strand below it, the reverse will be happening. Now here the DNA polymerase has to again move from 3' to 5' so that it can synthesize the new fragment from 5' to 3'. So here what happens is, in this part the replication is as we say is again lagging, because the till the time this replication fork will not open, the DNA polymerase cannot work smoothly. So a part of this will be moving this way, then if this new will open, then again it will move this way. So here the lagging strands are formed, and here because it will open and only one primer is required, and here it keeps on moving. So here we say that this is the leading strand. So overall, if I have to say that what happens inside a replication bubble, so if this is your replication bubble, so here will be your OIC, and from here the direction is in a way that here it is the continuous leading strand, but here it is the lagging strands. Then here it is again the lagging strands, and here it is the continuous leading strands. And in this way, the complete replication will take place. Now we have to learn about the various enzymes which are involved here. That is, we have to learn about that what all elements are there which will participate here in the reaction. So just give me a second. Yes. Okay. So now we will talk about the elements involved in the prokaryotic replication. Means here we have to talk about all those proteins which are involved in the replication of the prokaryotes. So one of the most important proteins which comes the very first here in all of this is the helicase protein. The helicase protein is the one which will help to open up the strand. So here we will require the DNA helicase, and we have already learned about the DNA B helicase. So this DNA B helicase will come to open the strand. So it will form the replication bubble. So the opening and the unwinding of strands will be with the help of this DNA B helicase. Second important protein here that comes is DNA gyrase, and DNA gyrase is the one which will help to remove the torsional strain. So it is also referred to as topoisomerase, and this topoisomerase will be helping to remove all the torsional strain which is produced while the DNA strands are opened. Then there is another important such enzyme which is SSB protein, and this SSB protein is single stranded binding protein. So while these strands are open, they are single stranded. So this SSB protein will particularly bind here so that this strand does not coil upon itself. Because right now this DNA strand is single stranded, right? So it can coil upon itself. So in order that it does not coil upon itself, and in order to prevent the secondary structures formation, the SSB protein is required, and it is also called as the single stranded binding protein. It binds upon the single strand of DNA once it is opened. Then comes our most important uh, enzyme, or the most important protein, and that protein is called as DNA. Means this is now the primase protein. So now it will be DNA G primase. Now this is the one which will be recruiting the primers on the DNA strand. So now the recruitment of the primers will take place, and how is it going to do? We are going to see it very soon, but please remember that the primase will recruit the primers on the DNA strand. After that comes the DNA polymerase 3, and this DNA polymerase 3 will do its replication activity as well as the proofreading activity. So from 5' to 3' it will do its replication activity, and in meanwhile time, if some wrong nucleotide is inserted or incorporated, that time from 3' to 5' it can also do its proofreading activity. And at the last, when the process is about to over, the whole replication when it is about to over, that time the DNA polymerase 1 comes, and this DNA polymerase 1 will help to again do the proofreading very accurately, and this DNA polymerase 1 can do the proofreading in 5' to 3' direction as well. There are certain endonuclease and exonuclease activities which DNA polymerase 1 has. So because of this, it can do very efficient proofreading as compared to DNA polymerase 3. So these are some of the very important proteins, and there are some important proteins like in E. coli. If I talk about, there is TUS protein. This TUS protein is the one which will help to stop the replication, or at last it will say that the replication should now stop. So it will come and bind to the strand so that the DNA polymerase now can no longer function. So this is how the whole of the enzymes are required here, and these are all those enzymes which are required during the replication process. So this I have told you, and I have already told you that the replication process in the prokaryotes will be occurring bidirectionally, and the whole of the prokaryote is monorepliconic. That is what we have learned. Now we will talk about the DNA G primase, because in our previous video I talked about DNA B helicase and the DNA gyrase in SSB proteins, as simple as that they will just help to stabilize the particular loop that is formed. So now I will move on to how this particular thing happens. So I will mark it in black. Suppose this is my loop form. Okay. Now here our whole of the activities will take place. So suppose I make my ORIC over here. Suppose this is the part. Wait. Now what happens is, this strand, obviously we know that this strand will act like what? This strand will act like in 5' to 3' direction, and similarly 5' to 3' direction. And this will act in opposite direction, 3' to 5', and here also 3' to 5'. Now what will happen? The protein which we have to talk about is right now is DNA G primase. Now DNA G primase has to recruit the primers. So I'll take another color for this. Let's take this color. Okay. So I was talking about DNA G primase. The main function of DNA G primase is to recruit the primers, and it itself is a kind of RNA polymerase. Please remember, its functioning is very similar to RNA polymerase. So when it makes the primer will have the RNTPs rather than DNTPs. So these RNTPs will be incorporated in the primer because of DNA G primase. And where will this primer be added? This primer will be added in the 3' end of the template strand. So here if I talk about, the primer will be in this way, like this. And here it will be the primer's 5' end, and here will be the primer's 3' end. Now we know that 3' end consists of the free OH group. So here also this primer will have free OH group, and this OH group will be definitely of those RNTPs only. Now what happens is, similar primer can be inserted over here also. See here what happens is that the fork is not opened. So this is only the maximum point up to which the fork is opened. So let's say that here the primer is inserted and it is in the direction again of 5' to 3'. Then here also we will have the primer inserted and it will be in the direction of 5' to 3'. And here the primer will be inserted here, because before this the fork is not opened, so here the primer cannot be inserted. So here also the primer is inserted from 5' to 3'. Now here the OH is exposed, here the OH is exposed, and here also the OH is exposed. Now comes the role of DNA polymerase 3. So DNA polymerase 3 has to now recruit the nucleotides, that is deoxyribonucleotides, just after this. So DNA polymerase 3 will recognize this OH of the primer. So this OH of the primer will be recognized by the DNA polymerase 3, and as soon as this OH is recognized, the DNA polymerase 3 will start recruiting the nucleotides here. So as the nucleotides are recruited here, this will become the leading strand, and here only this much part can be recruited. Then when the fork opens further, then this part will be recruited. So here the lagging strands are formed, and vice versa for this half. So this can be said as right half of replication fork, and this is the left half of the replication fork. So now let me draw how it will be. Here the synthesis of the nucleotides and the formation of the strand will be in this way, and this will be the 3' end, and here it will be like this way, right? So here also it will be 3' end, then here it will be again 3' end, and here it will be 3' end. This way the blue colored ones have shown that how the DNA polymerase 3 will now work. Now this DNA polymerase 3 has done what? It has synthesized the strands on all the four parts. Now this DNA polymerase 3 is a very important protein because it is recognizing this particular OH part, and then after this OH only it is starting to add the nucleotides. Now in the meantime, while it is adding nucleotides and forming the new strand, if one of the nucleotide is wrongly incorporated, then this DNA polymerase 3 also has the proofreading activity. And I have already told you that it has the exonuclease activity in the 3' to 5' direction. So because it has the exonuclease activity from 3' to 5' direction, it will do what? It will actually cleave that particular nucleotide and will again insert the right nucleotide. So this is what is the function of DNA polymerase 3's exonuclease activity. So this is what is happening here while the polymerization and the replication is taking place. Now there is another model, the similar model only, which suggests that while the DNA polymerase is functioning, now you see what is happening here. I'll try to show it again here so that it becomes very crystal clear to you. Suppose this is our particular strand, and here also we have the particular strand. Okay, now what was happening is the DNA polymerase 3 was in this way. We already know that here we have the beta clamps. We know the beta clamps, and after that we have the core DNA polymerase onto it, and upon the core DNA polymerase we have the tau protein or the dimerizing protein. Now the similar thing will be happening here. Here also we will be having this beta clamp. Upon this beta clamp will be the core DNA polymerase, and on this core DNA polymerase will be the tau protein or the dimerizing protein. Now these two dimerizing proteins interact with each other in a way that this DNA polymerase 3 here and this DNA polymerase 3 here work together. Now you might be confused in one of these statements here. I told you that they will work together, but how can they work together? Here it has to move this way and here it has to move this way. So don't you think that this DNA polymerase will move here and this DNA polymerase will move here, so this will be broken? So for this, there is a model which is suggested which says that this particular loop is turned in a way that it is looped in a way that the both DNA polymerase can go together and do the replication part. So what it means is that one loop is very normal. One loop is like this. The other loop turns in a way, suppose in this way it has been turned. So now what happens is one DNA polymerase is here, one DNA polymerase is here. Both of them are moving in this direction, but the only difference is here we are forming the leading strand. Okay, this is the wrong figure. And this loop cannot be turned, sorry. If this loop is turning, then we can form it like this way. Means what I'm trying to say is that this loop, if I'm trying to turn this loop like this way, then both of those DNA polymerase will be moving in the similar direction. But here in the above case, what we see is that the lagging strand will be formed, because you already know that here the lagging strand formation takes place. So here we'll have the lagging strand and here we'll be having the leading strand. So this loop forms so that the two can dimerize together easily and then they can work together. So this is how the functioning of DNA polymerase take place. For your kind information, if you don't remember the structure, here is the structure of DNA polymerase 3, and I already told you that there are the beta clamp units, then there are gamma complexes which helps to open and close these clamps, then there is a core DNA polymerase, and then there are the tau units which are the dimerizing units. The core DNA polymerase has two functions. One of them is the polymerization activity, and the another one of them being the exonuclease activity, and I already told you here also. Now all of this has happened. Now what next? What after this? So Let's see what will happen after this. Wait, I should draw it in black color. Okay, now in this way, suppose I'm drawing it with red. Here was what? Here was our leading strand, and this leading strand move in this way such that it synthesizes this strand completely. Okay. Let's not make it like this. Hmm. So from here, our leading strand started like this way, and it had lead to the formation of the complete continuous strand. And here had the formation of the lagging strands. And here in green color, I am saying that from here this leading strand was very continuous, and here we had the formation of lagging strands like this. Now this whole of the structure wherein more or less the whole of these are formed, this is called as the catenin structure. But this catenin structure has a problem. This is the hypothetical model which we are seeing, but you only try to imagine it from your view. It's just like the two rubber bands which were started to be uncoiled. They have been uncoiled, but at the point, there will be a point where these two bands will be fully uncoiled, but this part will still remain coiled. And this will be remaining coiled because this cannot be opened. Now, how to open this? To uncoil it and then again recoil means we have to basically cut this so that the two strands become free, and then we have to again recoil it. So now how can we do this? So basically here there are two particular enzymes which come, one of them being the DNA gyrase, that is the DNA topoisomerase, which I already talked about, and the second one is the topoisomerase type 4. These two enzymes, what do? They do is they cut these strands here and then they induce the negative supercoiling here so that the strands can get relief. Definitely they also consume the amount of ATP to do this activity. So they will first break the phosphodiester bonds and then they will again rejoin them, and in this way they will induce the negative supercoiling so that the catenin can get relaxed. Now all of this has happened and now we have finally got this structure. Okay, now there is one problem still, the remaining last problem. We have numerous primers, if you remember. Here also we had the primers, and then here also we had one of the major primers. And these all primers, as we already know, were made by whom? They were inserted by DNA G primase. Now what is the problem with these primers is that these primers are made up of RNTPs, that is ribonucleotides. These are the ribonucleotides, and the ribonucleotides are the characteristics of RNA. They cannot confer the stability to DNA. So we have to remove these RNTPs and we have to insert the DNTPs at their place. So what we need to do is that these RNTPs have to now be removed, like this has to be removed, this has to be removed, and in that way, instead of that place, we have to insert the DNTPs. So that what happens is more or less we can get the stabilized structure with no RNTPs. So this main function is done by DNA polymerase 1. So DNA polymerase 1's one main function is to remove all of the RNTPs and to insert in the place of RNTPs the DNTPs. And why DNTPs are required? So that the stabilization can be achieved. Also we know that there are in the lagging strands, there are these lagging strands which are formed on both of these strands in some or the other halves. Now what to do of those lagging strands? Now those lagging strands are called as the Okazaki fragments, and these Okazaki fragments also have to be joined. So they will also be joined later on. The gaps will be filled so that in any future case these should not be remain separated. So here the Okazaki fragments will be made continuous, the primers will be removed, and instead of primers the DNTPs will be incorporated, and all of this will be done by DNA polymerase 1. So the main function of DNA polymerase 1 is to remove the RNTPs or to remove the primers. Now it is upon you and you have to search it. It is your question that who is the one who is going to seal these Okazaki fragments? Is it the DNA polymerase 1? Okay, let me only answer this question. So do you think it can be DNA polymerase 1 who can seal these Okazaki fragments? There are the gaps in between and the gaps have to be ligated. Now this much amount of hint I have given. So I think and I hope that you can now go on to this query and you can try to solve this query by yourself. So this is how the replication takes place in the prokaryotes, and this is the circular mode of replication. In the next video, we'll talk about the regulation of the replication in prokaryotes. So that's all from my side for today. Thank you.